Right, good afternoon. Welcome to a Cadlam Cymru recording for A-Level Biology. My name is Mr. Davis, and I'm going to be doing a video on the sexual reproduction in humans part of the A-Level. Okay. Bear with me, my first language is Welsh, so I may stumble over some words on my spelling, not me, maybe not as good as it could be. Okay. Here is a list of all the Cadlam Cymru recording. I've just found a spelling mistake right there. Cadlam Cymru recordings for this spring session. Okay, so there's going to be 2 on cell structure and microscopy. There's going to be 2 on classification, both at AS level. Then there's going to be 1 for Unit 3 to do with respiration and chemoosmosis. And then the Unit 4 ones are going to be plant and human reproduction. Okay, so all of those will be available in this spring session of Cadlam recordings. Okay. Right, the session aims for today is to recap the subject knowledge for the 4.1 human reproduction unit. I'm also going to go over some past paper questions as well in order for us to apply the knowledge that we've recapped and learnt. Okay, if you have a preferred note-taking method, that's absolutely fine. Please carry on with that. But if you don't, I'd like you to try this method, the Cornell method. Okay, so you split your paper up like so. Okay, and in this 2/3 of the page, you put the main points. Okay, in your own words, abbreviations. You can put diagrams, mind maps, things there. Okay. Be efficient. Don't write down everything you see or hear. Leave out some of the the connecting words, things like that. After then the lesson, okay, you fill this bit in with cues and reminders. Okay, so how can I remember this bit? Well, you need to put something in there to help you remind. Any questions that you might have about it, bits that you don't understand, okay, they need to go there. And then again afterwards, you need to summarize what you've learned here. Okay, and these summaries make a really good start for any cue cards, flashcards is what I'm trying to say, flashcards that you might want to do before the end of the unit. Okay, so I'd like to try and keep up with that type of note taking for this lesson. Okay, cool. So the unit for 4.1 sexual reproduction in humans, it appears to be quite a short unit because you only have 4 sentences in the syllabus. Okay, but there is an awful lot going on here. Okay, it's a large unit with lots of different things to learn. Okay, so again, when you're doing your revision, okay, check your revision against these 4 sentences. Okay, and to check your understanding and make sure that you are in the right ballpark, okay, that you know what you're doing and that you are hitting these sentences in the syllabus. Okay, right, okay. Other resources available out there. Okay, the WJC website is a really good place to go. Key documents, okay, they've got the specification there, so that's the specification in detail. The teacher's guide goes into a little bit more detail if you need that. There are past papers and mark schemes on that website as well, really good. Resources as well. Okay, the WJC also do the blended learning. Okay, so if you Google WJC blended learning, okay, and then go to biology and you go to A-level, there's basically an online walk you through type of resource there. Okay, that's really good again for recapping the unit. Yeah, if you've missed a lesson in school or something like that, those are really good resources. Okay, there are also lots of other resources out there. Okay, so this biology mad one is fairly good. Don't get bogged down in, oh, I found this new resource, I found this new resource, I found this other resource and this other one and this new one. Okay, stick to 2 or 3 good ones. Yeah, it's better to have 2 or 3 good ones, i.e. these, rather than finding 40,000 ones that are a bit rubbish. Okay, you waste more time going through the resource than actually learning from it. Okay. So these are three resources that are really good to use, and obviously Carol and Cymru videos, they're obviously excellent as well. Okay, so the first part of the unit is basically structure and function. Okay, so this is a matter of learning. Okay, so if you stick this diagram, okay, in the first, in the 2/3 of your page, yeah, and then there's labeling. Okay, so you need to make sure that you get that labeled correctly. All right. So sometimes called the urinogenital system. Okay, so urinary as well because you're connected to the urinary system. Okay, so this is the urinary bladder connected there to the ureters. Okay, that you've learnt about in unit 3.7. Okay. Two testes. Okay, so they are the testicles. All right, where sperm are produced. Okay, then you have a structure on the back of that called an epididymis. Okay, and then there are folds inside the testes called seminiferous tubules. Okay, seminiferous tubules. So it's to do with semen. That's where semen production occurs. Okay, then you have a tube that goes from the testes all the way up around here, and that is known as the vas deferens. Okay, so the vas deferens is a tube that leads from the testis to the base of the penis. Okay, so when people have a vasectomy, okay, it's that tube there that is that's tied off, basically. Okay, so vasectomy means that you are unable to so the sperm won't get down the penis and cause fertilization. All right. Joining into the vas deferens there is the seminal vesicle. Okay, so the seminal vesicle produces seminal fluid. Okay, so we'll talk about what the seminal fluid does in a minute. And right there at the base of the penis, okay, you have this gland here that sits just underneath the bladder. That is the prostate gland. Okay, so the prostate gland just sits at the base of the penis there. Okay, and then the tube that the vas deferens joins that goes down the center of the penis is called the urethra. Okay, there we go. Okay, so those are all of the structures that you need to learn. Okay, you need to be able to identify. Okay, so each of those needs a function. Okay, so the scrotum, which you can't see on this diagram. Okay, so that's the sac that holds both testes inside. The sac is external. Okay, the reason the sac is external is that sperm production happens better at 35 degrees, which is why it's external to the body, which is at 37 degrees. Okay. Okay, the testes produce gametes. Okay, so they produce sperm cells. Okay, they also produce testosterone. Okay, so we'll talk about the hormonal cycles later on, but testosterone is a hormone that produces the male secondary sexual characteristics. Okay, do with hair growth and things like that. The epididymis, so those little structures there, that's where sperm cells mature. Okay, so maturation of sperm cells occur here. The vas deferens carries the sperm carries the sperm from the testis to the base of the penis during ejaculation. Seminal vesicles, as the name suggests. Okay, so seminal as in produces fluid. Okay, so it produces seminal fluid, so secretes fluid into the vas deferens, makes up over half of what the semen is, what is actually ejaculated. So that fluid contains lots of nutrients that enable the sperm cells to survive. Okay, so the sperm cells are able to take nutrition in order for respiration, in order to survive their long and arduous journey. The seminal fluid is also slightly alkaline, which helps neutralize any acidity that may be left in the vaginal tract after any urine. The prostate gland, again at the base of the penis, underneath the bladder, that also secretes a fluid into the vas deferens. Okay, prostate fluids contain zinc and ions. Again, helps neutralize the acidity that may be in the vaginal tract. Okay, so both of those glands here, glandular tissue, produce seminal fluid. Okay, so semen, which is what is ejaculated, is a mixture of sperm cells and seminal fluid. The urethra carries the semen, which is the sperm cells, spermatozoa, and the seminal fluid out of the body. Okay, also carries urine out of the bladder. Okay. And then the penis is a specialized organ adapted to transfer semen to the vagina during sexual intercourse. Okay. Okay, so you need to make sure that you've got structure and function nailed down for the male reproductive system. Okay. So you also need to do the same for the female reproductive system. Okay, so this is the view that we had here. Is a frontal view. Okay, the view that you have here is a side view. Okay, so this is the view from the side, not a frontal view. Okay, so you have an ovary here. Okay, but there are two. Okay, you can't see the second one because it's on the other side of the body. Okay, so that's an ovary. You then have a fallopian tube. So that fallopian tube is just here, and that connects with the womb. Okay, or the uterus, I should say. Really dates me a little bit. Okay, so the uterus is what we're calling it these days. So the uterus is the womb. It's basically where the embryo will implant and where the fetus grows. Okay, the lining of the uterus is called the endometrium. Okay, and some of you may have heard about endometriosis. Okay, so that's a condition where the endometrium goes into overdrive, basically. Okay, the cervix is not very obvious here, but the cervix is a ring of muscle that is at the base of the uterus. Okay, so that ring of muscle is very strong, and that will maintain pregnancy. Okay, basically it's the gateway to the uterus. Okay, that then joins onto the vagina. Okay, so the vaginal passage here joins out onto where the vulva is. Okay, so that's just the passage that goes up towards the cervix and towards the uterus. Just in front of that, okay, here you have the bladder. Okay, so that's the bladder just in front of the uterus. Okay, so pregnant women often need to go to the toilet quite often because they have a large baby growing in here, and that puts pressure on their bladder. Okay, so both of those tubes empty out into the vagina. Okay, so the vagina, into by the vulva, just there. Okay, so this grey thing here is the spine. Okay, and that is the pubis bone, the front of the pelvis. Okay, there, and this here is the colon. Okay, now obviously they're not part of the reproductive system, but they are on this diagram, just so you can kind of get your bearings. Okay, now you've probably seen a picture of the uterus or the female reproductive system that kind of looks a bit like this. Okay, so you've got, well, not like that at all, much better than that, but you've got two ovaries here, yeah, and then you've got the fallopian tubes leading to the uterus. Okay, and then you've got the cervix that then leads into the vagina. Okay, so the vulva and the labia are going to be the external parts of the reproductive system. Okay, So that's a strange diagram. It's a side-on diagram, okay? But you need to be able to identify those pieces. Okay, so the ovary, again, production of gametes. There are two, okay? So the gametes in this case are going to be secondary oocytes, okay? Try not to use the word eggs, yeah? Birds lay eggs. Mammals, and in particular humans, produce secondary oocytes, okay? They also produce hormones, okay? So there are similarities there with the testes, yeah? They produce the gametes and they produce hormones, okay? The fallopian tubes, or the oviducts, okay? Duct is another word for just tube, and ovi as in ovary, okay? So the fallopian tubes, or the oviducts, they connect the ovary to the uterus, okay? And they have a lining of ciliated epithelial cells. So they are the cells with little hairs on top that wave backwards and forwards, okay? And they waft the secondary oocyte down into the uterus. Excuse me. So the uterus then is a muscular structure that holds the baby during pregnancy. The endometrium there is the innermost layer of the uterus wall, good blood supply, builds up every month, part of the menstrual cycle. Okay. If implantation occurs, the endometrium is maintained. Okay. Cervix, ring of connective tissue, ring of muscle. That's the barrier, that's the gateway to between the uterus and the vagina. Okay. And the vagina, muscular walls, and it opens at the vulva. Semen deposited in the vagina during sexual intercourse, and then that will swim up through the cervix into the uterus, and the vagina is basically the birth canal. Yeah, that's where the baby passes through during birth. Okay, so again, structure, function, this is the bit, that's what it does. This is the bit, that's what it does. Okay, so you need to make sure that you are up on that. Okay, the next bit can be quite confusing, okay? So the next bit is gamete formation. So we're going to do spermatogenesis, so the formation of the spermatogonia, or spermatozoa rather, and then we're going to do oogenesis, the formation of the secondary oocyte, okay? So this occurs in the testes, in the seminiferous tubules we've spoken about, okay? And each testis has about 1 kilometre of seminiferous tubule. Okay, so there are a lot of these guys being produced. So the germinal epithelium is a cell in the wall of the seminiferous tubule. Okay, so if you can imagine, this is a cross section of the seminiferous tubule. Okay, I've got my kilometre of these. So this is a cell from here. Okay, in the wall of. So it undergoes mitosis. Okay, so these germinal epithelium divide to produce spermatogonia. Okay, or a spermatogonium, which is singular. Okay, and they divide many times through mitosis, and at the end of that, they form primary spermatocytes. Okay, so these here are primary spermatocytes. Okay, so spermatogonia growing here. Okay, and primary spermatocytes, they've divided and divided and divided. You can imagine all these cells along this wall here dividing, and they produce these cells. Okay, then there's a meiotic division. Okay, so that primary oocyte there has the diploid number that goes through meiotic divisions to produce a spermatid. Okay, so by the time that's moving this way, so by the time it gets to the lumen of the seminiferous tubule, it then is called a spermatid, okay, and that has the haploid number. So 2n there, our 2n number is 46, obviously, and our n number is 23, okay, number of chromosomes, so diploid number, haploid number, okay, and then maturation occurs, okay, so then they mature, or they will specialize into spermatozoa, okay, so spermatozoa, plural, spermatozoan, individual. Okay, so then these are the spermatozoa that will form part of the semen that is ejaculated. Okay, and then this bit, so these are now in the middle of my seminiferous tubule, and then these will mature in the epididymis at the end. Okay, the piece on the back of the testis. All right, so that's quite nice and simple. I've got we've got several mitotic divisions, one meiotic division, and then maturation, and then that gives me a spermatozoan. Okay, so spermatogonia, mitosis, spermatocyte, meiosis, spermatozoan, maturation. Okay, so you link those together in your head. That will give you the sequence of events that produces the sperm cells. Okay. That's basically what I've said there. Okay, so this is the epithelium of the spurminiferous tubule. Okay, spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, spermatozoa. Okay, so that there, your primary spermatocyte, that is the one that now undergoes the meiotic division to produce these. Okay, and then they are develop into spermatozoa. Okay, so these will specialize. Okay, they'll get their tail, they'll get their acrosome at the end. Okay, also heading through the seminiferous tubule. So that, again, if that's the tubule like this is just a cross section of one piece. Yeah, like that. Okay, you do have running down here these Sertoli cells. Okay, so the Sertoli cells provide nourishment and protection against the immune system. Okay, because again, these are going to have a different genetic makeup to the organism, aren't they? Yeah, these are going to be haploid. These are going to be different genetically to these ones here. Okay, so they need some protection from the immune system because of that. All right. TS drawing. Okay, so you need to be able to identify these parts on a microscope drawing. That is showing several of them. Okay, so that's a cross section across several seminiferous tubules. So this is where we are. So that's the outline of the seminiferous tubule there. This is the lumen. Okay, so we have your germinal epithelium here. These are your spermatogonium. These are your primary spermatocytes, your secondary spermatocytes, and then they go to your spermatids. Okay, and then the spermatids will mature in the epididymis. Okay, you've got cells in between these seminiferous tubules, okay, called interstitial cells or Leydig cells, and these are the ones that produce testosterone. Okay, and testosterone causes that spermatogenesis to occur, as well as the secondary sexual characteristics I've spoken about before. Okay, so this is a picture of a sperm cell itself. Okay, and you can tell that's highly, highly specialized. Okay, so you know everyone knows the little tadpole shape, don't they? But it's so much more than that. You have the head, which contains the nucleus, which has the haploid number of chromosomes in it. You then have an acrosome, which is a which is full of enzymes that carry out the acrosome reaction. Okay. You also have a midsection, okay, that provides the energy. Okay, there's lots of mitochondria in here, okay, that provide the energy to move the tail. Okay, so the tail is produced from lots of microtubules, and it extends out, and when that moves, okay, the sperm cell becomes motile, and that will be able to swim, okay, and propel the spermatozoa. Okay, oogenesis is slightly more complicated. Okay, so you do have similarities, but there are also differences. Okay, so during oogenesis, that happens in the ovaries. Okay, you have a germinal epithelium. Okay, so the same as you do in the spermatogenesis. Okay, so that undergoes a mitotic division to produce oogonium. Okay, an oogonium or oogonia. Okay, so far that's fairly similar to what we've had in spermatogenesis, isn't it? Okay, so, and again, the ium ending, spermatogonium, oogonium. Okay, right. then what happens is you then have that divides and these become primary oocytes. Okay, so same as spermatocytes, they are oocytes. Okay, the oocyte will then undergo a meiotic division. Okay, so it undergoes meiosis 1, but instead of there being 4 haploid cells, what you get is the first meiotic division produces a polar body and a secondary oocyte. So the polar body deteriorates, goes away. Okay, then the secondary oocyte is held in like suspended animation. Okay, so it doesn't complete its meiotic division until fertilization. Okay, so upon fertilization, okay, or when it's ovulated, the second meiotic division will finish. Okay, so then there's a second polar body which deteriorates, but that will leave me with an ovum. Okay, and that ovum, the secondary oocyte is haploid, but the ovum then has been fertilized, and it will then regain the diploid number. Okay, so some similarities, some differences. Okay, so similarity is there's mitotic divisions to start with. Okay, there's a meiotic division, but this one is suspended at the end of meiosis 1. Okay, and then after ovulation, the second meiotic division will happen. Okay, an interesting point is that when a baby girl is born, all the primary oocytes that she will ever ovulate are already present in her ovaries. Okay, so all this business here, the mitotic divisions here, have already happened in the fetus, in the embryo. Okay, and then what happens is these oocytes will be held then, and then when puberty arises, this will start to happen. Okay, so there's a mitotic division there. Okay, so that is gametogenesis. Okay, so the production of gametes. So there's a spermatogenesis and oogenesis there. Okay, so I would like you to be able to fill in that Venn diagram there. So what are the similarities and what are the differences between spermatogenesis and oogenesis? Okay, so you can stop the video and carry that out if you would like. Okay, right. Next bit, the TS drawing of an ovary. Okay, so there's an awful lot going on here. Okay, so the oocytes I've just spoken about here. Okay, what happens is they have a layer of cells around them. Okay, and that produces a follicle. Okay, so a primary follicle is a primary oocyte that has a bunch of cells around it. Okay. So that follicle, sorry, will develop into a secondary follicle. Okay, so secondary follicle, all these cells are dividing, and then there's a part in the middle that fills with fluid, all surrounding this primary oocyte here. Sorry, yes, so the secondary follicle contains a secondary oocyte. So all the while, this is getting bigger and bigger. Okay, and then by the end, what you have is something called a Graafian follicle. So this is a mature follicle. Okay, so it contains a secondary oocyte, it contains this bag of fluid here, it contains these cells, okay, that surround the oocyte. Okay, and then ovulation occurs when this Graafian follicle bursts out. Okay, so it joins onto the wall of the ovary, bursts out, and then a ovulation occurs and the secondary oocyte with some of the cells is released into the fallopian tube. Okay, so what happens then is this goes on its merry way down the fallopian tube to either be fertilized or not. The remainder of this Graafian follicle here turns into something called the corpus luteum. Okay, why Latin I don't know, but corpus luteum, white body. Okay, That's what corpus luteum means. So development is stimulated by LH, okay? And if fertilization occurs, it starts secreting progesterone and estrogen. So this has a role in hormone production, and then what stems then from those hormones being produced. So during a woman's monthly cycle, okay, a secondary oocyte is going to be ovulated, but there's going to be a sequence of these Graafian follicles and secondary oocytes in secondary follicles and primary follicles all developing. Okay, so this is kind of like a little conveyor belt of secondary oocyte production. Okay, sometimes two secondary oocytes or two Graafian follicles ovulate at the same time. Okay, so that's where you get non-identical twins. Okay, so two different secondary oocytes being released, fertilized separately. That's two non-identical twins. Okay, so that's what's going on there. So again, on your notes page, you need diagram, structure, and function. What's the bit called and what does it do? So a secondary oocyte is what is ovulated. Okay, so this is the bit that's ovulated out of the ovary. Okay, so you have the oocyte in the middle here. Okay, and as you can see, it is, excuse me, the chromosomes in the middle there are held suspended halfway through meiosis 2. Okay, so there's an awful lot. So there are some things in this unit that you need to make sure you go back and revise 1.6 cell division for. Okay, so you remember the chromosomes, they join onto the spindle at the equator and then they are pulled apart during anaphase. Okay, but these chromosomes are held in a line on metaphase 2. Okay, right, so what we have around our oocyte is the zona pellucida. Okay, so this is formed when it's a primary secondary follicle. Okay, so that zona pellucida is a jelly-like structure, jelly-like coat around the outside to do with protection, and it does have a function later on. Okay, there are also cortical granules. These are granules that have chemicals in them that again have a job with the zona pellucida, and I'll talk about them in a second. Okay, you can see that arrow's in the wrong place, so this is the first polar body. Okay, so I've talked about meiosis occurring, and then there's the follicle, there's the oocyte, and then there's the polar body that breaks down. Okay, so that's one of them there. Okay, and then on the outside of that, okay, you have the corona radiata. Okay, so this is the remnants of the secondary follicle. Okay, so like I said, remnants of the Graafian follicle. Okay, so the Graafian follicle ovulates this. Okay, so this is your secondary oocyte. Okay, with some remnants of the secondary of the follicle cells around it. Okay, so during fertilization, okay, what happens is the spermatozoa have swum a colossal distance for a sperm cell. Okay, so sperm cell, smallest cell in the body, has a very large distance to travel from the cervix right up the uterus and down the fallopian tube. Okay, until it comes into contact with a secondary oocyte. Okay, so it's not just fertilization. Okay, so fertilization, there is a 4-step process. Okay, so first of all, there is capacitation. Okay, so what capacitation does is it increases the permeability of our cell membrane in the head of the sperm above the acrosome. Okay, so capacitation occurs in here. Okay, so the sperm starts to get close and this starts to break down. Okay, what you then have is the acrosome reaction. Okay, so the acrosome reaction releases enzymes, okay, which break down the zona pellucida. Okay, so this is the capacitation reaction. Okay, and this breaks down that jelly-like layer we were talking about before. Okay, so that's the acrosome reaction. Number 5, fusion. Okay, so fusion is the sperm and the secondary oocyte membranes joining together, and it's the genetic material entering the cell. Okay, so the membranes join on here. That haploid nucleus enters the cell. That triggers the completion of the second meiotic division to form the ovum and the second polar body. Okay, so the second polar body is going to go off down there. Okay, and then those two join together to make a haploid nucleus. All right. What happens then is, so as those two join together, what happens then is the cortical reaction. So remember the cortical granules we were just talking about before? These fuse with the cell membrane and they change the structure of the zona pellucida, which prevents any more sperm entering. Okay, so often students ask, oh, well, what if 2 sperm go in at once? That isn't possible. Okay, so when one sperm enters, that completes a chain reaction of second meiotic division occurs, cortical reaction, that changes, we're done. Okay, no more sperm can enter through. So those two nuclei form, they fuse together, and that produces the zygotic nucleus. Okay, so that nucleus there is the beginning of an entire new person. How exciting! Okay, so fertilization, 4 processes there. Okay, so there's intercourse, capacitation, acrosome reaction, fusion, cortical reaction. Okay, so that probably best as a list or as a table. Yeah, so a table as in, so the steps, so say step 1 is capacitation, and then what occurs at that step. Yeah, and then you've got fusion, what happens there. Okay, so that would be a good way of writing those notes. Right, so our ovum has been ovulated, it's been fertilized, which means we now have a zygote. Okay, so that zygote now begins its journey, it's beginning to be wafted by the ciliated epithelial cells down the fallopian tube. Okay, so 1 cell undergoes mitotic division, 2 cells, mitotic division, 4 cells, mitotic division, 8 cells, until it forms something called a morula. Okay, so a morula there is a group of cells, unspecialized cells, okay, that will develop into an embryo. Okay. For those of you that are interested, unit 4.6, okay, there's a bit about stem cells there. Okay, so stem cell research stems from things like this. So what happens is that morula, again, it begins to develop, and it will develop something called a blastocyst. Okay, so a blastocyst is, that's all those cells, they've all formed into a circle around like that. So it forms basically like a hollow ball. Okay, and this part here will develop into the embryo. Okay, and the rest of it will develop into the amniotic sac. Okay, so it will come down here and it will implant into the endometrium. Okay, so the endometrium is that thick layer of blood vessels, and that is where it's implanted, and that is where the placenta will join on. Okay, and then the implanted blastocyst will implant there, and it will grow out, and then the fetus, or the embryo to start with, and the fetus, and then the baby will be growing in the uterus. Okay. So again, 3 different types of 3 steps to implantation there. Okay, so cleavage, the zygote undergoes meiotic division to form a morula and then form a blastocyst. Okay, so this is a blastocyst. Okay, so the blastocyst has an inner load of cells, and then it has that circle, that trophoblast, the circle of cells on the outside, and then it has a blastocyst cavity, okay, and then it will join into the endometrium wall. Okay, so blastocyst moves into the uterus, it attaches into the endometrium. Cells on the outside of the blastocyst, the trophoblast, form trophoblastic villi. So these are going to form villi that go into there. Okay, they penetrate the endometrium. That increases the surface area so we can absorb more nutrients for the embryo. Okay. And that is where the placenta will begin to form. Okay, so this will develop into the placenta. Okay, the green cells there will develop into the baby, and the rest of it will be the amniotic sac. Okay. Okay, so the placenta and the amniotic fluid that develop around the baby. So there's exchange of gases and nutrients through the placenta. Excuse me. The placenta provides a barrier between the maternal blood and the fetal blood. Okay, so it protects the fetal blood from the higher pressure of the mother's blood. Cells of chorionic villi fuse together, preventing the mother's phagocytes from passing on to the fetus. Okay, so obviously now the developing fetus has a different genetic makeup to the mother, which means the mother's immune system would destroy it if it could. Okay, and that is the part of the basis for morning sickness. Okay, so the woman who's pregnant feels ill because her immune system's fighting against itself. All right, but the mother's phagocytes can't actually get to the fetus to destroy the cells through the placenta. And there's also hormones there that can maintain pregnancy as well. Okay, so that's the role of the placenta. It's another diagram showing you that. So this here is the trophoblast. Yeah, around there. That germinal disc developing into the embryo, connected to the placenta by an umbilical cord. Yeah, and that umbilical cord there has these. So these are the chorionic villi that grow into there. Okay, so it increases the surface area, short diffusion distance, countercurrent flow, some of the things that you've learned previously in order to increase exchange of substances. Right, hormones and the menstrual cycle. Okay, so this can be quite difficult. So you have 3 graphs there. Okay, 4 graphs, sorry. All right, so you have the graph at the bottom that is dealing with estrogen and progesterone. You have the graph in the middle dealing with LH, luteinizing hormone, and FSH. I've forgotten what FSH is. We'll find out in a minute. You've got the third one, which shows the ovarian cycle. Okay, so that conveyor belt I mentioned of the formation of an ovule. And then you've got the uterine cycle, which is basically the thickness of the cell of the endometrium. Okay, so the endometrium is thick here and thin here. Okay, and then it's across a cycle. This time we've got 28 days. Okay, so it is a cycle of about 28 days ish. Okay, obviously it varies from person to person. Okay, right. How to do this is to be able to recall that from memory, and be able to label it with all of these numbers. Okay, so that's just the way to do it. It's just to remember. So you can chunk it down into this hormone graph, this ovarian cycle, that uterine cycle. Okay, but it's just a matter of remembering what does what. Okay, so this is what each of those numbers represent. Okay, so I'd like you to draw those 4 graphs, okay, and then label the graphs with these functions. Okay, so the anterior pituitary gland secretes follicle stimulating hormone. That's what FSH stands for. Sorry, everyone. So the anterior pituitary gland in the brain produces FSH. That stimulates the maturation of a primary follicle to a secondary follicle. Okay. A maturing follicle then secretes estrogen. Okay. estrogen stimulates LH, it inhibits FSH, and triggers the rebuilding of the endometrium. Okay, so you can see we've got a relationship between all of these hormones. Yeah, they all have an impact on each other. So estrogen, having stimulated LH, stimulates the secretion of FSH, induces ovulation on day 14. Okay, so day 14 of the cycle is when ovulation occurs. Stimulates the conversion of a Graafian follicle into a corpus luteum. FSH inhibits estrogen production. Okay, so you've got a kind of cycle here. Estrogen produces FSH, which inhibits estrogen. Okay, so that controls the levels of those follicles. The corpus luteum, you'll remember that's left over from a Graafian follicle ovulating a secondary oocyte, secretes progesterone. Okay, and that progesterone maintains the endometrium, keeps it thick. Okay, it inhibits FSH and it inhibits LH. Okay, corpus luteum will also release some estrogen. However, during the second half of the menstrual cycle, the estrogen doesn't stimulate LH secretions because of the effect of the progesterone. Okay, so you can see there's a very complicated relationship between all four of these hormones, isn't there? Okay, falling FSH and LH cause the corpus luteum to degenerate. Progesterone levels decreases, the corpus luteum declines, and low levels of progesterone and estrogen cause the endometrium to break down and to be shed during menstruation. Okay, and that then will recur with number one. Okay, so it's a cycle. So post-puberty, that cycle happens roughly once a month until the menopause. Okay, so like I say, this 4 graphs here, okay, learn what each of the hormones does, okay, learn what they do pregnancy, menstruation wise, and learn what they do to each other. Okay, so again, whether that's the graphs, whether that's a mind map, whether that's a table, okay, but that's something you need to make sure you've learned. Okay, so following fertilization, the blastocyst begins to secrete human chorionic gonadotrophin, HCG. Okay, if the implantation is successful, the developing placenta will then start producing HCG. And HCG is important because it maintains the corpus luteum for the first 16 weeks. And you'll remember maintaining the corpus luteum has an impact on progesterone, which has an impact on the thickness of the endometrium. Okay. Corpus luteum secretes estrogen and progesterone, which inhibits FSH, inhibits LH, so it stops any more ovulating happening, stops superovulation. Estrogen stimulates the growth of the uterus to accommodate the growth of the fetus. Stimulates the growth of the mammary glands for milk production. Progesterone for wall of the endometrium, which I've spoken about, and suppresses the uterine wall to contract by inhibiting secretions of oxytocin. Okay, so we're going to talk about oxytocin during birth. So then during pregnancy, the corpus luteum degenerates and then the placenta takes over the production of those hormones. Okay, so just before birth, estrogen levels increase and progesterone levels decrease. Oxytocin is secreted, and secretions of oxytocin stimulate contractions of the uterine wall. Okay, and more oxytocin means more contractions. Okay, that's what a positive feedback system is. Okay, you remember a negative feedback system from the homeostasis unit. A positive feedback system, oxytocin produces contractions, contractions produce oxytocin. Okay, that's why contractions become close together, they become more often, and they become harder. Okay, the contraction becomes more forceful. Prolactin is also secreted, and that stimulates production of milk by the mammary glands for the baby. Okay, so again, there are some fancy bits beyond just the menstrual cycle. Okay, so if the menstrual cycle carries on, that's fine. If the menstrual cycle doesn't carry on and fertilization occurs, then this is the sequence of events. Okay. Okay, well, there was an awful lot going on there, wasn't there? Okay, so there's an awful lot to learn, despite it only being 4 sentences in the specification. Okay, so in order to apply this information now, okay, you need to answer some past paper questions. Okay, so recall and consolidation of subject knowledge is about 50% of what you need to do, but you also need to apply it to the exam questions. Okay, so the following slides have got a mixture of past paper questions for you to try and some peer marking exercise. Okay, so first one here, that's a nice big complicated one on the hormone cycle. Okay, so mammals have evolved different patterns of ovulation, which are controlled by the changes in hormone concentration and affected by copulation, which is a fancy word for sexual intercourse. Graphs show the changes in the concentrations of LH and FSH and progesterone during the menstrual cycles of a human and the rabbit. Okay, so using the graphs and your knowledge of hormonal control of the human reproductive cycle, explain the effects of hormonal changes which are shown, discuss the similarities and differences in the control of ovulation in rabbits and humans. Right, lots of students will quite merrily waffle away for about a page here, okay? Right, but this question is not a waffly question. Basically, the question is asking this. Okay, explain the effects of hormonal changes. So that is basically a list, isn't it? Yeah, this hormone does this, this hormone does this, this hormone does this, this hormone does this. Okay, right. Discuss the similarities and differences. So what is similar between this one and this one? What is different between this one and this one? Okay, and even be that systematic. Okay, so talk about what happens to the progesterone level here, talk about what happens to the progesterone level there, talk about what happens to the LH here, talk about what happens to LH here. Yeah, and doing a comparison makes it more difficult for the marker not to give you the mark. Okay, so I'd like you to stop the video now and I'd like you to have a bash at that question for me, and then when you have finished you can restart the video and I will show you the mark scheme. Okay, so mark scheme coming up now. Okay, so remember these types of mark schemes, it's not one marking point, one mark. Okay, you will need at least half of those to get into the middle band. Okay, but again, yeah, FSH does this, which does that, which does this. Yeah, so that is basically a learny bit. Yeah, you've learned that, you know what FSH does, you know what estrogen does, you know what LH does. Okay, so that's just that happens. Okay, so similarities, what's similar? Differences, what's different? Okay, good job. Give yourself a mark out of 9 for that one. Okay, and then we'll move on to the next question. Okay, so photomicrograph below shows section through a human ovary. Okay, so you know you straight away you're thinking of a Graafian follicle there, primary follicle here. Okay, there you go. Name the structures. Fine. All right. There's a nice mathsy question there. Okay. In the human female, the first half of the meiotic division takes place just before ovulation. The photomicrograph below just shows the surface of an ovary ovulation. I'd just like us all to take a minute and be amazed at that photograph. That's actually a photograph of ovulation occurring. That's pretty cool. Okay. So again, these questions, quite easy. What's X? Yeah. 2 outer layers of X. Okay, describes that. They're more learning questions. Okay, so stop the video and have a bash at those answers, and I will publish the mark scheme for you in 3, 2, 1. No, I won't. There's a more to the question, I'm afraid. Sorry. So state how the secondary oocyte usually prevents entry of more than one sperm. So there you're talking about your cortical reaction, aren't you? Yeah. Suggest a reason why there's an increased risk with IVF. So IVF isn't on the syllabus. The information for that is going to be here. Okay, so you need to dig around in the question and find that information. Developing embryo is not transferred to the uterus till 3 days after IVF. Use your knowledge of fertilization to explain why this delay is needed. Okay, so you think about that. Think about the significance of what happens in those first 3 days. Okay, and I'm going to show you the mark scheme now, honest, in 3, 2, 1. Okay, so again, you can stop the video and you can mark your question and its answers out of 13. Okay. Right, next one. State 4 differences between the composition of blood in the arteries and the umbilical arteries and the umbilical vein. Okay, so what's the difference between the blood in arteries and blood in veins? Yeah, that's a nice, easy one. 2 reasons why there must be a barrier. Okay, so what are we stopping getting into the baby's blood from the mother's blood? Okay, think about the morning sickness thing I said. The arterial blood flow to the placenta is high, and the blood in the intervillous space exchanged 3 times per minute. Suggest how the following features. Okay, so again, lots of exchange very quickly. Okay. Pressure in the uterine arteries is 10 times greater than the pressure in the intravillous space, and the length of capillaries in the placenta is diminished. Okay, so again, they're not common sense answers, okay, but you need to know about the structure, what the function of the placenta is, and what it's trying to do, and then link that to these features here. Okay, so again, stop the video and then you can, I will publish the mark scheme for you. Oh, there you go. Okay, so that's the mark scheme. So I would like you to give yourself an answer there. Okay. So again, nice learning questions in the top here. Okay. Okay, that's a learning stuff, and this here is more, you know, I know this, therefore. Okay. Good job. Okay, another one. Clomiphene medication used to treat some forms of infertility leads to increased release of FSH and LH. Suggest how that increased the chance of conception. Okay, so think about what those hormones do in order to increase the release of something else. Okay, prolactin, oxytocin hormones play a role in birth feeding. Prolactin concentrations stay higher in women who are breastfeeding. Prolactin inhibits the production of FSH. Describe the role of prolactin in women who are breastfeeding. Explain why they have reduced chance of conception. Okay, so prolactin, less FSH. What implications does that have? What does that mean? What does that mean? What does that mean? Okay. Explain why oxytocin production is an example of positive feedback. Okay, so like I've said, more of that means. Okay, so stop the video, have a bash at answering those, and I will publish the mark scheme in 3, 2, 1. There's the mark scheme for that. Have a mark of those and see how many marks out of 8 you got. Okay, so stop the video and give yourself a mark. Right, next little bit is peer marking. Okay, so this is a question we have already seen. You've already seen the mark scheme. I'd like you to have a read of Johnny's answers, and I'd like you to tell me how many marks you think he got. Okay, so stop the video, have a read, and see how many marks you think he got. Okay. So this is the actual marks that Johnny got. Okay, he was all right on the first one. He was all right on the second question, B part 1. B part 2 makes the womb contract. Okay, so that little doohickey there, that bit means he's missing something. Okay, so the womb wall contract, which releases more oxytocin, that increases. Okay, he got that one the wrong way around. Okay, so but he did very well there. All right, next one. So this is a question that you can have a go at. Okay, so stop the video and have a bash at that question. And I will publish the mark scheme in 3, 2, 1. Oh, 1 second, more to the question. So publish the mark scheme in 3, 2, 1. There we go. Okay. So stop the mark scheme and give yourself a mark out of 13, I think. Okay. Right, so Johnny's had a go at this question now. Okay, so you've seen the mark scheme. Okay, if you want to scroll back through the video to see the answers, that's absolutely fine. Okay, but you can stop the video and give Johnny a mark and see how many mark he got. Okay, and I will publish his answers in 3, 2, 1. Done it again, sorry guys. So more answers from Johnny there, so you can stop the video and 3, 2, 1. Okay, so he got the ovulation correct. Okay, so there's again words missing here. Okay, so terms missing. Okay, what were the terms that he missed there? Okay, so type of cell division, term given to the event shown at A. Okay, so cleavage, yeah, he's misunderstood. Okay, and again missing another term there. Okay, so you can see how term heavy, it's important to get the terminology correct for these answers. Okay, and he did very well there as well. Okay, so got 2 marks for C part 1 and 2 marks for C part 2. Good. Okay, so I think this is our last question. Okay, there's an awful lot of reading to do there, so you can stop the video and have a go at that. Second part of the question is there, so you can stop the video and have a read of that. And here are the answers for that question. Okay, so stop the video and have a read of that. Right, that was a mammoth video there. Okay, so there are just a few techniques there to help you practice and revise. Remember, lots of different ways of revising, so find one that works for you. And remember, 50% recall information, 50% note making, but 50% of your work needs to be past paper questions. Okay, excellent, guys. Thank you very much. Work hard, and I will see you again.